In this video, I'll be continuing to discuss changes to DNA. The first video was on mutation, and then on DNA repair, and then a subsequent one on infectious changes to DNA, where I discuss viruses and transposons. And in this one, I'll discuss introns and plasmids. So I include introns into this section, and introns are a bit unusual in that most of their function is to act as sort of buffers between two exons, but many introns actually serve different functions. The RNA actually becomes a controlling factor in expression. And it's clear that many introns have originated from transposons or retrotransposons. Some of them are still capable of moving from one place to another in the genome, or in converting one species into another species by inserting a copy of the intron into a particular location within a gene. There are four major types of introns. Group 1 and Group 2 are self-splicing introns. Group 1 are more like transposons, and Group 2s are more similar to retrotransposons. These also include Group 3, which are very similar to Group 2, and twintrons, which are essentially an intron inside of another intron. Then there are spliceosomal introns, which are primarily in messenger RNAs, and these are derivatives of Group 2 introns. And then finally, archaeal introns, which are different than the others, and there may be other classes or types of introns as well. The first introns were discovered by extracting RNA from the nucleus and extracting RNA from the cytosol, and then hybridizing to a specific gene probe. And what was found was in the cytosol, while there was only one band corresponding to that mRNA, there were several bands corresponding to that same RNA in the nucleus, and they varied in size. And it was eventually deduced that these were from different pieces of the RNA being sequentially chopped out, so that the initial RNA, called heteronuclear RNA, those were chopped out the introns were chopped out one by one by one. Initially they were called intervening sequences, but eventually were called introns to distinguish them from the exons, the expressed parts of the RNA. Other evidence came from electron microscope data, and in this case this is a technique called R-looping. And so DNA of a certain region was denatured and then allowed to hybridize to RNA for genes of the same region. And what was found was, in the case of mRNA, the mRNA was much shorter than the actual gene, and these little small loop outs and large loop outs were caused by intronic sequences. Also, other parts that did not hybridize to the DNA were the cap on the end, which was often difficult to see, but then the poly A tail at the end could be seen as an unhybridized region at the 3' end of the mRNA. From this information from the electron microscope, our looping figures, then you could draw a map of the gene with all of the exons, E1, E2, E3, etc., as well as the introns. And some genes have a lot more intronic sequences than they do in exonic sequences. Here are the three general mechanisms for Group 1, Group 2, and spliceosomal introns. In the case of Group 1, there's normally a conserved U at the 5' intron exon border and a conserved G within the intron at the 3' intron exon border. And then you can add GMP, GDP, or GTP, free guanosine, and that becomes covalently bonded to the 5' end of the intron, freeing the 5' exon. And then there's a hydroxyl nucleophilic attack at the other end at the 3' intron exon border, forming a free intron, and it can circularize in many cases, and then ligated exons. In some cases, the intron can contain a transposase type gene, which is an endonuclease that cuts out specifically a region of a chromosome, and actually it can get itself inserted, which is similar to how a DNA transposon inserts itself. Group 2 introns, on the other hand, have an internal adenosine near the 3' intron exon border, which affects the first reaction of splitting the 5' exon off and creating a lariat-like structure of the intron. And then there's a similar second reaction which ligates the two exons together and frees the intron. In some cases, group 2 introns carry along with them a reverse transcriptase, and they also can transpose similar to a retrotransposon. Spliceosomal introns look similar in their mechanism to a group 2 intron, except they rely on spliceosomes, which are RNA and protein modules that fit together. Some of those look somewhat like group 2 intron structures, and so it's clear that they are derivatives of a group 2 intron. And their splicing mechanism, other than having these spliceosomes that are attached to them, is similar to the mechanism for a group 2 intron. Here is some of the secondary structure shown for group 1 introns. Often many of these are found in the ribosomal RNA locus of fungi and sharks and a whole bunch of different organisms. We've been working with these for several decades now. But in B down there, you can see the structure of the secondary structure of these introns, including P regions, pairing regions. P1, P2 are just looped regions. P3 is the interaction between one part of the molecule and another, and then P4 and 5, and then P6. P7 is also distantly paired between two different sections of the RNA, and that's the region that holds on to the free guanosine. And then there's P8, P9, and P10. The larger arrows there show the 5' intron exon border and the 3' intron exon border. More recently, people have been looking at this as far as the functional regions of the secondary structure. So in D, I've cut out C, so that's not shown here, but in D, you can see on the left-hand side the P4, P5, P6 regions of the intron, and those are called the scaffold because those hold the other parts of the intron in position. P1 is shown in the middle because that's where one of the splice sites is, and then the P7, P8, P9, and P3 regions are the catalytic regions. The P7 is the part that holds the free guanosine, and then the P1 region and the P10 region are brought together in the middle of that molecule for the splicing reactions. This is a slightly different view of the same introns, group 1 introns, and you can see the dark lines there are the 5' exons and the 3' exon. And then the P7, you have a special GC pair, which is where the guanosine is held, which is the first part of the reaction. Then that guanosine attacks the 5', 3' border, freeing up that end of the intron and also freeing the exon. The triangles on here, the little O's, are where open reading frames can sometimes be found, and those usually encode an endonuclease. The diagram on the right is the alternate view of the intron, and this shows more of a functional view, so you can see how the two exons are brought together in this form. Again, the P456 constitute the scaffold. The P1, P10 regions are where the exons are joined, and then the P7 is where the free guanosine is held for the first part of the reaction. Again, you can see the little triangles with O's in them. Those are open reading frames, often encoding a transposase, which is an endonuclease in this case. Here is just another view of that, showing the P456 as a scaffold with some conserved regions indicated in purple, and then P1 is in the middle with the special GU pair, which is where the intron-exon border is. Then there's the G site in the P7 region, and then up where P9 is, it loops around to ultimately go to this omega G, which is the last G in the intron, which is where the intron-exon border is in this case. On the right-hand side, joining the P7 and P8 regions, this J8/7 region, is an area of fairly high sequence conservation, which is unusual for these group 1 introns, or introns in general. Structures are conserved, but sequences aren't. So whatever this sequence is and why it's so well conserved isn't yet known yet. This structure, the same color scheme is used as in the previous figure, and it shows how the P8 is sticking out there. P3, P7 you can see, which is holding the free guanosine, is in the middle there, and the intron-exon border would be right in the central portion of that molecule. Group 1 introns are self-splicing in vitro, although the process is more efficient and speeded up in vivo by chaperone proteins that hold it in its catalytic form. But in vitro, you can add guanosine, and that'll attach to the P7 region. Then you can attack at the 5' intron-exon border. The guanosine becomes covalently attached. Then the two exons join together covalently, freeing the intron. In our work with introns in fungi, we found a set of very small introns in the ribosomal locus of several different fungi. And instead of being hundreds of nucleotides in length, they were only between about 60 and 80 nucleotides in length. And they appear to have only a P1 and a P9 region, but the P9 acts like a P7, so that's where the guanosine is held. And they splice in vitro as well as in vivo, even though they are quite small. However, even though they did splice in vitro, they needed help. So we cloned a piece of the ribosomal locus out and then put it into expression vectors to make RNA to test different regions to see whether we could get in vitro splicing. If we used just the intron, no splicing occurred. If we included the intron plus part of the 5' region of the small subunit, also no splicing occurred. But when we had the intron as well as the little end of the small subunit SSU and the internal transcribe spacer 1 ITS1 and a little bit of the 5.8S gene, it did splice in vitro. That piece of RNA did splice in vitro. We then tried to mutagenize different pieces of that cloned sequence, and we did that by just synthesizing DNA primers for polymerase chain reactions and then made new clones with the mutation. So on the right hand side, any part that is listed in red, if that was mutagenized or changed, that means that abolished any sort of splicing. If it's green, that means that if that part was missing or mutagenized, it had no effect on splicing. So you can see in this case the intron plus part of the ITS region, which is in red there, was necessary for splicing, although we don't know how it helps splicing in this case. But I think it's clear that other pieces of the ribosomal locus were responsible for folding the intron into an active conformation. Group 2 introns are also self-splicing, although in vivo they also rely on proteins to increase the efficiency of the splicing process. They have several pairing regions, but they also have domains 1 through 6, D1, D2, D3, D4, D5, and D6. D5 is actually fairly well conserved for an RNA sequence. It's found in almost every group 2 intron and is pretty much characteristic of them. There are the dark lines down there are the 5' exon and 3' exon, which pair up. The 5' exon has internal binding sequence 1, IBS1, and IBS2, and those pair with external binding sequences EBS1 and EBS2. There are also other regions that pair up alpha and alpha prime, beta and beta prime, etc. And then in D6, you can see the adenosine, the A sticking out there, which is the nucleotide that starts out the reaction at the 5' intron exon border, covalently bonding to the other part of the exon or the intron to form a lariat structure. The triangle with the O in it, often there is a reverse transcriptase found in that spot, so this is why they look similar to retrotransposons. And the structure and form of the reverse transcriptase is very much similar to other reverse transcriptases in mitochondrial retrotransposons and other retrotransposons. So as I indicated earlier, this is a self-splicing group 2 intron, self-splicing in vitro. But the start of the reaction is initiated by an internal adenosine, which then attacks the 5' intron exon border. You get a splitting off of the exon and formation of the lariat. And then the 5' exon reacts with the 3' exon intron border, splicing those together, those two pieces together, and freeing the lariat. And it's very characteristic. You can actually see these lariats in the electron microscope. Some of the group 1 and group 2 introns are mobile. So on the left-hand side for group 1 introns, the ones that are mobile usually encode for an endonuclease, and the endonuclease recognizes a particular spot on the DNA, cuts that, and then you get an insertion of a copy of the intron. In the case of group 2 introns, on the right-hand side, they encode several proteins, including a reverse transcriptase and an endonuclease usually, so they'll hit a target sequence. They insert the RNA into that spot, and then the reverse transcriptase and DNA polymerase finish the process by making a DNA copy of the intron in that location. In the middle, there are also some things called inteins, and these are actually proteins which can be spliced. So instead of splicing the RNA, they splice the protein, but they can also make a copy of the intine and insert it into a particular location in the chromosome, and they do that again with an endonuclease that hits a target sequence for those. Spliceosomal introns splice in a very similar way to group 2 introns, except for the fact that they rely on snRNPs, SNRPs, which are small nuclear ribonuclear proteins. SNRP1 binds to the 5' exon intron border, and SNRPU2 binds to the A nucleotide region. And so the reactions are similar, although they rely on these particles that have RNA and proteins together. And if you look at the sequences of the UN, U2, etc., they resemble regions that you would find in a group 2 intron that are integrated into the intron itself. In a few cases, they're also related to ribosomal RNA regions, and so it could be a process that's common to both introns and ribosomal RNAs. Archaeal introns are quite different from the other introns, although they do have possibly a common origin. So on the end of some tRNAs, as well as some ribosomal RNA regions, these introns are often found. And they're found primarily in the anticodon loop, but as the little triangles indicate on the left-hand side, they can occur in many, many different places. In this case, there's a structure in the intron itself, which is a bulge helix bulge. And so you can see there's not much sequence similarity, but the structure is the same in each case. And an endoribonuclease, not an endonuclease, but an endoribonuclease cleaves this, and then the ends fuse together to form the mature tRNA. In this case, the anticodon is within this region also. During evolution, these introns may have provided some flexibility in altering the genetic code at those anticodon sites as well. When you look in archaeal ribosomal RNA loci, as well as the loci in some other organisms, including bacteria and eukaryotes, you find that when they are processed, the ribosomal RNA is processed, it does so in a manner that's similar to an archaeal intron splicing, in that there's a pairing and you get a bulge helix bulge region, and then you have clipping of those sequences, freeing up the small subunit ribosomal RNA and the large subunit ribosomal RNA, which circularize, and then those are further processed by other RNAs to become the mature small subunit and large subunit ribosomal RNAs. And in this case, there's a tRNA involved there as well. The triangle O in this case also indicates the location of the possible endoribonuclease. If you look at the small subunit and large subunit genes for a large number of ribosomal RNAs in a wide variety of organisms, you find introns located in many different portions of each one of those genes and RNA molecules. So in red here are group 1 introns, green are group 2 introns, blue are spliceosomal introns, and black are archaeal introns. Looking at which organisms they're found within, as well as the extensive nature of being found in many, many different portions of these genes and RNAs, it's clear that they've been involved in the ribosomal locus, probably in its evolution, for a very long time. Also, if you look at the evolution of RNAs involved in translation in general, the small and large ribosomal RNAs seem to have been joined together in pieces, so that you have preformed pieces being added on, similar to adding on, Lego blocks one to another. And I think, and I wrote in a paper, that these may have been joined together by introns. So the introns have been in the ribosomal genes for a very long time, because they helped to put the ribosomal RNA together. Likewise, in the tRNAs and mRNAs, the introns have been important in the evolution of those molecules, putting pieces together as well to form functional units and to probably accelerate the speed of evolution of those. If you compare the positions of those introns, red being group 1, green group 2, blue is spliceosomal, and black are archaeal, and you look at the different phases in the evolution of the ribosomal subunits, both the small subunit and large subunit, you find that one of the first types of introns that were there were group 1s, followed by group 2s and spliceosomals. And if you look in the catalytic center of the large subunit, specifically, only group 1 and group 2s are in that very ancient piece of the molecule. There are additions of group 2s in phases 2, 3, and 4, followed by decreases, and increases in spliceosomal introns in phases 5 and 6. In many locations on these two molecules, both the small subunit and the large subunit, there appeared to be overlaps or very close proximity between group 1s, group 2s, and spliceosomal and sometimes archaeal introns. In fact, sometimes they looked like they were in exactly the same spot or close to the same spot, within 10 nucleotides. And especially this location here, where we did a little study on modeling of the introns and what they look like, because at first we thought it was a group 2 intron, or possibly a spliceosomal intron, and then later we thought that it also had characteristics of a group 1 intron. So we did a little modeling and came up with a surprise, actually. So in this one location, if you take the same sequence, then you can actually form that sequence into a group 1 conformation. Makes a perfectly good group 1 intron. But if you fold it in a slightly different way, it looks like a perfectly good group 2 intron, including having the conserved D5 domain as well as the unpaired A in domain 6. In each case, all you have to do is move the intron-exon border over by one or a few nucleotides, and you can have it be spliced as a group 1 or alternatively as a group 2 intron. This had me wondering about the evolution of these introns and whether they were related or not. Often it's thought that they aren't related, but I think that they may be related and that they're evolving and have been evolving in the ribosomal region for a long time. So in an RNA-centric world, you could get these different pieces of both the tRNAs and the ribosomal RNAs joining pieces together, either by insertion and integration or just end ligation accomplished by the introns. Now in the ribosomal region in a DNA-centric world, then you can start with a group 1 intron, which seems like the initial type of intron that was that evolved first. And then you can get duplication, movement into a new locus, either by transposition or some other mechanism, and movement into a different locus, or moving out into the mRNAs, for instance. But then you have this evolution, essentially, of a group 1 intron becoming a group 2 intron, just by moving the border around and having a different construction of that intron. Those can also duplicate and then move to new locations. The group 2s can become spliceosomal by losing some of their internal structure and relying instead on spliceosomes, which are produced, which are RNA sequences, some from introns and some from ribosomal RNA genes, which makes sense since it looks like they've been evolving from the ribosomal region. This process can be accelerated by increasing the number of ribosomal DNA copies, and so now you have speeded up evolution as well as, and then you can get transposition from there into different ribosomal loci, ribosomal RNA loci, as well as into mRNA loci, and by increasing the numbers, and most eukaryotes have tens of thousands of copies, or at least hundreds of copies this process of evolution of the introns now can be accelerated. The spread of the introns can also be accelerated by endosymbiotic events, such as moving an alpha proteobacterium into an archaeal cell where it has carried group 1 and group 2 introns. Cyanobacteria carried group 1 and group 2 introns into the archaeplastida, as well as into the rhizaria, and then these have spread out in different ways into stromatophiles, alveolates, Acrobia excavata, amoebozoa, fungi, and animals. Some of those have come directly from either bacterial cells or archaeal cells, and others exist within the organelle still. But certainly the movement and transfer of these introns has been extensive over a period of billions of years. The inclusion of introns in mRNA genes and other genes has been quite successful, and that's because it increases the rate of leading to new combinations of genes and parts of proteins. So recombination in genes without introns is a little bit dangerous, because if you get a crossover and you shift over by 1 or 2 nucleotides, then it destroys the reading frame and might destroy the function of that protein. Now, if you have an intron in that region and you get a crossover, it's much less dangerous because the reading frame is not destroyed. You can still splice those together, those exons together, and still have the same functional reading frame. Many of the sequences within an intron, it doesn't matter if they're mutated in one direction or another. The intron still can be functional. So in addition to reducing the chance of destroying a gene by changing the reading frame, so that now you can get different parts of 2 different alleles joined together in different ways, you can also create new possibilities. That is, you can create genes with different portions of different proteins on the lower part here. So you can have a gene of function A, gene of function B, get a crossover between them, and now create 2 new alleles for those types of genes. Possibly, for instance, one might be a transmembrane protein that now becomes a protein that winds up in the cytosol, and vice versa. And so that can change quite a bit of the gene structure. Or you can have a phosphorylation domain that switches from one protein type to another. So again, essentially, you can create new classes of genes that weren't there before, thus accelerating some evolutionary processes. It also allows the alternative splicing process to originate and evolve. And that's where now you can have one gene not producing just one protein, but a single gene can then produce multiple proteins having different functions. So there are some where you have standard splicing, or ones where you skip exons, mutually exclusive exons. You can have ones with different 5' ends or promoters, different 3' ends, polyadenylation sites. Introns can even be used as part of the coding sequence in some cases. Also, exons can be split so that part of the exon can be used as part of the message, and other parts can actually be spliced out. So you can have quite a few different possibilities of making new types of genes by alternative splicing schemes. So far we've talked about viruses and how they're related to transposons, and how transposons are related to introns, and how each one of those have contributed to evolution and evolutionary processes. Now I just want to finish up by talking about plasmids, which have some similarities to viruses, as well as some similarities to bacterial cells, genomes, and genes. Plasmids come in several forms, and they're just categorized primarily as autonomously replicating sequences. That is, they're not on the main chromosome of the organism, but they're separate replicating pieces of DNA. In the case of bacteria, often they're circular. They have replication genes, an origin of replication, conjugative transfer genes in some. In others, a transposon, including antibiotic and heavy metal resistance. And they often are retained by the organism primarily because they have those antibiotic or heavy metal resistance genes. Some larger plasmids, such as the TI plasmid in Agrobacterium, have an origin of replication, of course, and then some genes that encode for plant hormones, which will then change the plant cells to form a callus or some other tissue based on what those particular genes are doing. They also have virulence genes, which allow them to infect certain tissues, cognitive transfer gene or conjugative transfer genes, and these look somewhat like genes for a virus, for viruses to move from one cell to the other. And in this case, only the auxin gene, cytokine genes, as well as opine genes and some others are transferred on a piece of DNA called the tDNA, which also resembles infection by a viral nucleic acid. And that actually gets integrated into the plant chromosome. Some of the plasmids carried by fungi are linear rather than being circular, and they often have proteins covalently bonded to the ends, and those are the black dots on the ends here. Some carry a toxin gene as well as an antitoxin gene for immunity to that toxin. So if the organism is carrying the toxin gene, it can kill cells without the toxin genes unless they also have the plasmid, because they have the antitoxin gene, which confers immunity. So this thus propagates or continuously propagates that particular plasmid. Plasmids and DNA in general can be shuttled from one cell to the other by conjugation, that is, attachment of the two membranes together and followed by transfer of the plasmid DNA, or transformation, just pieces of DNA that can be taken up by the cell, including plasmids and other sequences, and by transduction, that is, transfer into the organism via viruses. Plasmids that carry antibiotic resistance have several mechanisms for getting rid of the antibiotic. One of the ways is to produce an enzyme that degrades the antibiotic, and a second way is to produce an enzyme that alters the antibiotic so that it no longer can kill the cell. And then a third way is to produce efflux pumps that pump the antibiotic out of the cell. Agrobacterium, which infects plant cells, carries along with it a TI plasmid, which can transform plant cells. So when it infects a plant cell, it sends the tDNA from the TI plasmid into the plant nucleus, and that DNA, the tDNA, incorporates into the chromosomes of the plant. This produces hormones which cause the plant cells to act abnormally, and then they form a gall called a crown gall. Here is a diagram of the TI plasmid from Agrobacterium tumefaciens. You can see it has an origin of replication, so it can replicate in the bacterium. It also has a conjugative transfer set of genes which allows it to move from one bacterial cell to another. And then it has a virulence region over here, which once it contacts a plant, it senses that, turns on the genes of the virulence region, and those genes then set up a system so that a copy of the T region is transferred into the plant cell. Now the T region has a left border and a right border, so it looks sort of like a transposable element, but it also moves from the bacterial cell to the plant cell in a way that makes it look sort of like a virus as it moves over. It then moves to the plant nucleus and transfers those genes into the plant nucleus, and those are plant hormones. And the plant hormones then affect cell division and the shape and form of the cells, forming that crown gall or callus tissue. Here is a diagram showing some of those processes. So there's the TI plasmid. Plants form certain compounds, phenolic compounds, that then stimulate the TI plasmid to start producing the virulence factors, which then form tDNA, copies of the tDNA region, which is then coated with a protein, and then the contact is made between the bacterial cell cytoplasm and the plant cytosol, and the tDNA is transferred into the cytosol, then into the nucleus, and then it integrates into the chromosome of the plant cell, where it starts producing the plant hormones to cause cell division, callus formation, and eventually forming a large gall of that tissue. So this process employs a plasmid, but the plasmid then acts somewhat like a transposable element, or at least parts of it act like a transposable element. And then because it coats the nucleic acid with a protein, it looks somewhat like a virus as well. So we started by talking about viruses and how they're related to transposons, and how transposons are sort of reduced versions of viruses, and introns are reduced versions of those, although there are crossovers among all those. And then we were just talking about plasmids, including the TI plasmid, that has some characteristics of viruses, as well as some of transposons and plasmids. So there's sort of a melding of some of these processes for some of these mobile genetic elements. In all cases, they've led to changes in the genetic material and acceleration of some evolutionary processes and the evolution of different groups of organisms. This is the last video on changes to DNA, where first I discussed in the first video DNA mutation, second video was on DNA repair, and then a third one on infectious changes to DNA, including viruses, then one on transposons, and then this final one on introns and plasmids. So that pretty much finishes up this section. Thanks, and I'll talk to you later.